Hi Hires, we are looking at the Metabolism and Survival Unit, proteins in membranes. Okay, so some revision and maybe a little bit more on the membrane. So you should know that it's a fluid mosaic model, and the fluid is made up by phospholipids. And what we have with phospholipids is a particular type of molecule called a hybrid molecule. So one end behaves one way and the other end behaves another way. So at the top we have the head, and the head is hydrophilic, meaning it loves water. And at the base we have the tail, and the tail is hydrophobic, meaning it hates water. Now if you're doing higher chemistry, this can all be explained in terms of polar and non-polar, and if you're not, you just need to know it's loves and hates. Okay, so what happens here is that since the inside of the cell is pretty much liquid, water, and the outside of the cell pretty much water as well, okay, with things dissolved in it, but basically a solution, all of the heads want to be on the inside and the outside. All of the tails want to be nowhere near the inside or the outside. So they form a bilayer with all of the tails pointing in the way and the heads on the outside. The mosaic part, we should again already know, but just to be absolutely clear on what this is. So the mosaic is made up by proteins. The proteins are either moving all the way across the membrane, okay, so like this one here and this one here. These are integral proteins because they're going across the entire membrane. On the outside or inside, ones that are stuck to the membrane but are not going all the way across are referred to as peripheral proteins. And if you want to change the membrane, you just change what's there. So those peripheral proteins and the carriers and the channels and everything that goes into the membrane will decide what that bit of membrane can do, what it can respond to, and what it can then do as an activity, whether it's to send on another signal or whether it's an enzyme, but you can alter what the membrane is. A reminder, diffusion is the movement from a high concentration to an area of low concentration, and you can do that as long as they are small, just straight through the membrane. So oxygen, carbon dioxide, they can just get straight through. However, some of them are a bit big. So if you want to move them, what you need, still from a high to a low, and you're still not going to use any energy, but you're just going to give them a hole that is exactly the right size and shape so that you can move them straight through. Okay, so this is called facilitated diffusion, and it will either be by a channel, which is just a hole, or a carrier, and the carrier will actually move it. Okay, so as well as having holes just with particular channels, we also can use proteins as pumps which are moving against the concentration gradient. So these ones require energy, this is active transport, and this is from your NAT5. But the reason we need to have them is a little bit more this level, which is why we use them to maintain concentrations in compartments, I'm saying here, but that could be the whole cell if necessary. The best example is probably the sodium-potassium pump. You don't need to know the exact detail. The cytochrome carriers, which are part of the electron transport chain and respiration, you do need to know a lot more detail about them, and also are in photosynthesis, so it's worth you knowing those. So as well as your carriers, just a reminder, we can use proteins for a lot of different things in a membrane. So they could be signaling, they could be holding it to different places, we can use them as enzymes. And as you pick which particular ones you have, that will decide what that particular stretch of membrane can do. The ones that you need to be able to give, they are pores, so that's your channels, they are pumps, so that's normally for active transport, are they enzymes. Examples would be nice. Make sure you're happy with those three.